Paper electrophoresis. Here, in paper electrophoresis, what is mean by electrophoresis? Electrophoresis is a separation technique where the ions will be separated out. Okay, in this video we will discuss about the paper electrophoresis. Okay, in this paper electrophoresis we can study in this topic. In today class we will discuss about the introduction, principal components of paper electrophoresis, nothing but paper, electrodes and electrolytes, and the types of electrophoresis, that is nothing but low voltage or high voltage or horizontal or vertical. Factors which what are the factors which governing the migration of ions in this paper electrophoresis? We will discuss about this advantages and disadvantages of the paper electrophoresis and some applications. Okay, let's begin our video. Then what is mean by paper electrophoresis? Here in paper electrophoresis, it is a separation technique where the ions of different charges are separated on a medium of a paper by application of a voltage between two electrodes. First of all, we have to get the paper, and with this contact with the two electrodes, the electrodes will having voltage, and the ions will be get separated, which are on a medium. Okay, here the ions of different charges are separated on a medium of a paper, which is also called as moistened with a buffer by the application of voltage. By the application of voltage, the ions will be separated each sides by giving some contact. Okay, in this paper electrophoresis, it is a simple separation technique where the ions of different charges are separated on a medium of paper by the application of voltage between two electrodes which are in contact with the paper. The paper electrophoresis is primarily used to separate the mixtures of amino acids, proteins and peptides. The main principle of the paper electrophoresis is separation technique. Okay, this is a separation technique. Here a mixture of ions are applied on a center of a paper which are immersed on a buffer solution, that is called as ionic strength, and this paper is placed across the two trays then which are filled with a buffer into the two electrodes are immersed. When a voltage is applied across these electrodes, the ions migrate towards to the anode and cathode. According to the charges, they will move, means ionic substance are moved towards to the anode and cations will move to the cathode. Ultimately, there is a separation of anionic and cationic, and also non-ionic substances will be separated out. To detect these ions, we have to use some sprays or reagents to get the visualized that paper chromatography. Both qualitative and quantitative analysis are performed in this paper electrophoresis. Here, the electrophoretic mobility, that is the migration of ions, that will be based upon the charges of particles, size and shape of the molecules, and voltage, pH, ionic strength, buffers and dielectric constants, and some viscosity of a medium. These all will be factors, main factors which affect the electrophoretic mobility. And the paper electrophoresis used to separate an environmental factors such as nothing but temperature. If the charge on the molecule is more, the migration is faster. You can understand? Okay. If the charge on the molecule is more, the migration is faster. If the charge of the molecules is less, then migration will be slow. Bigger molecules are migrate slowly when compared to the smaller molecules because they having smaller atomic size, so they will be fastly moved to the upper region. This is about the principle of the paper electrophoresis. We have completed the introduction and principle. Now we have to see the components of electrophoresis. What are the components? Paper, electrodes and electrolytes. Which type of paper is used in this electrophoresis techniques means here in this paper electrophoresis, paper is used as a supporting medium. When we compare to the gel electrophoresis or capillary electrophoresis, in that types, the agarose gels and polyacrylamide gels, starch blocks are used as a medium for the separation of the ions. But here, in this paper electrophoresis, we are going to use the Botman filter paper, which is having 2.5 centimeters length and 5 centimeters width. The paper strip will be touches on the buffer solution which kept the as electrode vessels. The paper is used as a washed with distilled water, which is having followed by the 0.1 molecular HCl, that is nothing but hydrochloric acid. To remove the impurities, we have to clean that Whatman paper with the hydrochloric acid. When we move to the electrodes, here the electrodes are used which are in the form of thin wire, okay, that is made up of carbon or platinum. These type of electrodes are used. In low voltage electrophoresis, the voltage across two electrodes is about 10 to 300 volts. And in high voltage electrophoresis, the potential is about 50 volts to 215 volts per centimeters. When we move to the another one, third one, that is electrolytes. Here, the electrolytes are depend upon the different types of pH. To adjust the pH and ionic strength, we have to use some buffers. Here some types of buffers are used, which are some types of buffers are given. That is nothing but main three types, that is barbiton buffer, tris acetate buffer, citrate buffer. These three types of buffers are given in this topic, and to this the components are also completed. When we move to the types of paper electrophoresis, here the paper electrophoresis is two types based upon the voltage, okay, high voltage and low voltage. In high voltage paper electrophoresis, the separation is very fast and sharp bonds are obtained, and sharp bands will be separated closely to the compounds, and more number of samples can be analyzed by the high voltage paper electrophoresis. And when we see in the low voltage electrophoresis, in this low voltage electrophoresis, we can see the horizontal type and vertical type, okay. You can see here a diagram which is a horizontal and vertical, okay. These are the paper electrophoresis which is vertical in shape. This is a vertical type paper electrophoresis. In this electrophoresis, the buffer solution of pH and ionic strengths are filled with the beakers, and Whatman filter paper is placed in between the two electrodes within the solution, and in this vertical mode, the migration of ions will be assisted by the gravity, okay. The separation will be takes place in this vertical mode of paper electrophoresis is in about 6 to 8 hours. After sufficient migration, the paper is taken out and dried. Then we observe the spots, okay. When we move to the horizontal one, here you can see the horizontal paper electrophoresis. This is the diagram view of here some main factors which are nothing but charges of ions, size of the ions, viscosity of the medium, voltage, and pH. These are the main factors which are mainly affected the migration of ions in paper electrophoresis. There are some factors. There is 5 types of factors are affected to migration of ions in paper electrophoresis. That is nothing but charges of ions, size of the ions, viscosity of the medium, voltage applied, and pH of the buffers. These are the 5 main factors which affecting the paper electrophoresis. And when we came to the advantages and disadvantages of the paper electrophoresis, 3 main advantages are there. They are: this technique is very easy to follow and low cost instrument, and here only paper and buffers are used to separating the migration of ions. Okay, and when we came to the disadvantages of the paper electrophoresis, here the separation time is more when we compare to the gel electrophoresis. Here the separation time is more, so that's why it will be time taken. And high voltage is in these 2 types of voltages are using, that is high voltage, low voltage. In high voltage it will be cause dangerous, so these are the main disadvantages. And applications, when we came to the applications of paper electrophoresis, this paper electrophoresis is mainly used for the separation of amino acids, proteins, and lipoproteins in serum, and separation of the enzymes which are present in the blood, and 5th one is separation of alkaloids. Nothing but antibiotics, which are different samples. Okay, this is about the paper electrophoresis. Thank you so much for listening my class. Thank you very much.